Hello students and welcome to the lecture of Microbiology paper 1 Introduction to Microbial World. We'll be studying chapter 2 Golden Era of Microbiology and the topic for today's lecture is Robert Koch's contributions in microbiology. In this lecture we will be studying the germ theory of disease, the Koch's postulates, the staining technique for tuberculosis and identification of the tubercle bacilli, as well as the cholera infection. So we'll just see the introduction of Robert Koch, how he developed as a medical student and a doctor. Robert Koch was born on 11th December 1843 in Klosthal, Germany. He was the son of a mining engineer. He gained interest in science while he was in school and he decided to study biology. He chose a career in medicine while studying at the University of Göttingen, but natural sciences was always his passion. Koch developed a strong interest in pathology and infectious diseases as a student of medicine. When he was a student, he won a research prize for his studies on neuronal innervations of the uterus. He graduated from the university in 1866. Later, he served as an army doctor in the Franco-Prussian War. But after the war, he moved to Poland and established a successful clinical practice in Poland. He investigated the anthrax disease in 1876. In 1882, he investigated the tuberculosis disease, the causative agent of tuberculosis, and in 1883, he investigated the cholera infection. He also formulated the Koch's postulates, and in 1905 Robert Koch won a Nobel Prize in Physiology or Medicine. So before we go on to the germ theory proposed by Robert Koch, we'll go to the history of the germ theory. In 1530, Girolamo Fracastoro, a physician and scholar in Venice, published a poem, Syphilis Sive Morbus Gallicus. In this poem, he described the shepherd Syphilis who had the disease which has since come to be known as the syphilis. In 1546, Girolamo Fracastoro recognized the contagious nature of diseases. He considered contagions to develop from three different sources, that is contact, fomites, and from a distance. But for the lack of proof of his ideas did not have much influence during this period. In 1835, Agostino Bassi, Italian in Lodi, showed that the silkworm disease, muscardine, was caused by a parasitic fungus which was later named as Beauveria bassianea in honor of Agostino Bassi. Later in 1843, Oliver Wendell Holmes, poet and physician in the US, wrote a story on the contagiousness of the puerperal fever, in which he pointed out that a woman who had just been delivered should not be attended by a physician who was conducting autopsies or attending patients with puerperal fever, and that the puerperal fever could be thus conveyed from patient to patient. Holmes also stated that washing the hands in calcium chloride and changing clothes after attending a patient with puerperal fever were preventive measures. These ideas met with fierce opposition by the established obstetricians of the day. In 1850, Ignaz Semmelweis, a Hungarian physician who was an assistant in the first obstetric ward of Vienna Hospital, where the puerperal fever rate was exceedingly high. He observed that students came to this ward directly from the dissecting room without washing their hands. In the second ward, where midwives received instructions, much greater attention was paid to cleanliness, and therefore the puerperal fever rate was very low in this ward. This observation, as well as other pieces of evidence, convinced Semmelweis that puerperal fever was a septicemia. So Semmelweis enforced washing of hands in calcium chloride, and by this very simple measure he brought about a dramatic fall in the rate of puerperal fever. But he was subjected to mockery or ridicule, and this disrespect contributed to the insanity and the early death of Semmelweis. In 1861, Semmelweis published the paper The Cause, Concept and Prophylaxis of Childbed Fever or the Puerperal Fever. Later, in 1854, John Snow suggested that cholera was likely to be transmitted via the accidental ingestion of the fecal matter from an asymptomatic individual. In 1865, Louis Pasteur isolated the parasite causing the pebrine disease from silkworm. He also showed that the disease could be eliminated by the use of healthy or disease-free worms for breeding stock. In 1867, Joseph Lister, a British surgeon, introduced antiseptic surgery by the use of phenol to soak the surgical dressing. This conquest of wound infection was his great contribution in the field of microbiology and medicine. In 1875, Jacob Henle, who was Robert Koch's anatomy teacher in Göttingen, was defending the idea of contagium animatum, that is, disease could be caused by living transferable entities. Later in 1876, Robert Koch, the German physician, was the first person to demonstrate the relationship of a specific organism with one type of disease, and by proving that Bacillus anthracis was the etiological agent or the causative agent of anthrax. He proposed the germ theory. That is, the germ theory was in existence from long time, that is from the 1530, where Girolamo Fracastoro described the disease of syphilis, but the people did not have proper proof to show that the disease was caused by a specific organism. Thus, the germ theory was a very hot debate in that time, and the role of bacteria in the cause of diseases was uncertain. So Robert Koch began to investigate the anthrax disease. Right? And as he was the medical officer in Poland, he was given the work to find the details about the anthrax disease that had killed around 528 people and over 56,000 livestock. So, in order to study the anthrax disease, Robert Koch started observing the blood samples of the infected animal, and he found the presence of rod-shaped structures in the blood of infected animals which was not present in the healthy animals. So he was sure that the rod-shaped bacteria was the cause of the disease. He isolated the bacteria from the blood of the sheep which had died because of the anthrax infection. He grew the cultures isolated from the blood of the diseased sheep in the laboratory in pure culture forms, and he injected the purified organisms in the mouse. After injecting the organism in mouse, the mouse produced similar results, symptoms as the sheep. He repeated his process for over 20 generations in the mice. And finally, in 1876, he announced that the rod-shaped bacterium that was present in the blood of the infected animal was the causative agent of anthrax. Robert Koch propagated the organism in his laboratory in order to study the life cycle of the organism. And during his studies, he observed that the organism, it elongated, and after elongation, by the process of fission, it propagated. He observed that growth of bacteria under optimum conditions was that the bacteria would grow, elongate, it would swell and lead to the formation of long filaments of the organism. In the later stages, the filaments would acquire granules which later developed into the refractile spheres. Still later, the filaments decomposed, but the spheres they remained in the medium. Robert Koch observed that bacteria should be present in order to cause the disease or to transmit the disease. So he developed certain artificial culture media, and along with development of artificial culture media to cultivate the organism, he also developed sophisticated techniques in microbiology in order to observe the organisms. He also observed that when the fluid was dried, the organisms did not appear to be present in the fluid or in the dried material, but the spheres remained in the dried material. So Koch hypothesized that these spheres were nothing but the spores of the bacteria, and that these spores could cause the disease. The formation of the spores by the organism were responsible for the contamination of soil, and thus the soil could remain toxic for a number of years because of the presence of the spores. Pasteur also noted that there were certain pastures in which the cattle grazed contained the spores of the bacilli, due to which the cattle suffered from the disease. And because of the presence of the spores of Anthrax bacilli in the soil of the pastures remained poisonous for a number of years, and therefore avoiding the grazing of cattle in specified pastures was sufficient to avoid the disease in the cattle. So, the germ theory is basically based on the concept that many diseases are caused by infections with microorganisms, and that and Robert Koch was the first to link the specific bacterium with the specific disease. Based on the work of Louis Pasteur and the germ theory of disease, Robert Koch established the basic scientific requirements used to demonstrate that each specific disease is caused by a specific microorganism. These requirements were based on Koch's experiments with the anthrax organism that was isolated from the diseased animal, and therefore these postulates are known as the Koch's postulates. The Koch's postulates consist of 4 rules. The first important rule is that the microorganism must be present in all the individuals that are affected by the disease, but the organism should not be present at any time in the healthy individuals. So the first rule states that the organism should be present only in the diseased animals and not in the healthy animals. The second rule is that the organisms should be isolated from the diseased individual and should be grown in pure cultures in the laboratory. That is, every time it should be possible that the organism should be isolated and cultivated in pure cultures in the laboratory. The third rule says that the cultured microorganism should cause the disease when it is introduced into the healthy animal. The organism that is grown in the laboratory, when it is injected into another healthy animal, it should also cause the same disease. And the fourth important rule is that the microorganism must be re-isolated from the experimental host and found to be identical to the original organism. That is, after you have experimentally infected the animal should show the symptoms, and after the symptoms are seen in the animal, the similar organism that was isolated in the second rule should be isolated from the experimentally infected animal. So it can be demonstrated that a diseased animal whose blood sample contains the bacteria that is responsible for the symptoms of the disease, isolate the bacteria from the blood and grow it in pure cultures in the laboratory. The laboratory culture is inoculated in the healthy animal. The healthy animal should show the symptoms, and the bacteria which was isolated in the step 2 should be isolated in the step 4, that is from the experimentally infected animal. While a healthy animal should not show the presence of the bacteria at any time in the blood. So these are the postulates, and from this postulates Robert Koch established that a specific organism is responsible for a specific disease in the animals or humans. After studies on the anthrax organism, Robert Koch started studies on tuberculosis because in Europe, tuberculosis was responsible for 1 out of the 7 deaths. Preliminary studies on tubercle bacilli or tuberculosis had established its transmissibility, but the causative agent was not yet found. Suspecting that the tuberculosis was caused by bacteria which are not stained by the conventional stains, Robert Koch experimented with new stains. He used the Ehrlich's methylene blue stain to stain the organism, and he detected a few tiny rods in the tuberculous tissue. When he added a brown counter stain for photographic contrast, he observed more bacteria. He then noticed that the old stains were more effective than the fresh stains, and he hypothesized that the old stains had absorbed a useful chemical, mainly ammonia, from the air, which made the methylene blue stain alkaline. So, he observed that alkaline stains are more effective than the freshly prepared stains. So, Robert Koch began adding caustic potash to his stains to achieve similar effects as the old stains. Thus, he developed a stain for the bacterium, although it was not very effective for visualizing the slender rods, but a method for staining was devised. Paul Ehrlich had described the acid-fast properties of the bacterium. Acid-fast property is the property of the bacteria or the ability of the bacteria to resist decolorization with acid alcohol, which confers acid fastness to the bacterium. And therefore, conventional stains could not be used for staining the tubercle bacilli. In the 1890s, Frederick Nelson and Franz Zeil modified the conventional stain by adding phenol, that is carbolic acid, and basic fuchsine in order to stain the acid-fast bacteria. The acid-fast bacteria have a very high concentration of mycolic acid, that is a lipid, in their cell walls. And because of the large concentration of the lipid, the property of acid fastness is conferred to the bacteria. The high amount of lipid, which is a waxy substance, prevents the entry of the stain in the bacterial cell, and thus the bacteria are not easily stained. But once the bacteria is stained, because of the waxy substance in the cell wall, the stain cannot be easily removed from the bacterial cell, and thus the stain which was modified, now named as the Zeil-Nelson's carbol fuchsine stain, which has got its name from the ones who devised the technique, Frederick Nelson and Franz Zeil. So Zeil-Nelson carbol fuchsine, because of the addition of phenol, which is nothing but the carbolic acid and the basic fuchsine. So this stain could easily stain the tubercle bacilli. And thus, after staining the tubercle bacilli with this modified stain, Robert Koch discovered the tubercle bacilli. So after the discovery of the tubercle bacilli, Robert Koch further tried to isolate and cultivate the bacteria in the laboratory in pure cultures. In order to grow or isolate the bacteria, instead of using the liquid broths, he used the solid media for growth of the Mycobacterium tuberculosis organism. He used solidified cow or sheep serum, with sterilization being obtained by repeated short courses of heating at 58 degrees Celsius and finally prolonged heating at 65 degrees Celsius, then solidifying the medium, which was poured in slanted tubes so as to increase the surface available for inoculation. This also to, permitted the tubes to be kept for weeks without contamination, although it contained a rich medium for the cultivation of Mycobacterium tuberculosis. After inoculation, the tubes were incubated at 37 to 38 degrees Celsius and growing colonies were detected by the naked eye. A total of 15 pure cultures were obtained from 4 experimentally infected guinea pigs, 4 from bovine tuberculosis, and 7 from patients. Robert Koch noticed that detection of growing colonies using a 30x or 40x magnification allowed the detection of colonies by the end of the first week. Otherwise, it would take 10 to 14 days for the visible colonies to appear on the medium. After experimenting with different media at different temperatures, Robert Koch found that he could grow colonies on coagulated blood serum at 37 degrees Celsius. So 37 degrees Celsius, that is the body temperature of a human being, was the optimum temperature for the growth of the bacteria. Later, to test whether these bacteria grown in the laboratory could cause the disease similar to the disease that was caused by the naturally occurring organism, he inoculated around 217 animals with the bacilli from pure cultures that he had grown in the laboratory. All the animals that were inoculated with the pure culture of organism showed the formation of tubercles, and the number of tubercles that appeared in the animal was proportional to the size of the inoculum that was injected into the animal. So, Robert Koch concluded that the tubercle bacilli was the cause of the disease. Later, Koch detected the bacilli present in the sputum of the individual. So he found out that he could induce the disease in healthy animals by inoculating them with the infected sputum. Thus, he concluded that the sputum was the principal source for the transmission of the disease. Later, in 1882, Robert Koch presented his findings on tuberculosis in a meeting of Berlin Physiological Society. In his demonstration, he included more than 200 microscopic preparations of the tubercle bacilli and presented it to the public in the Berlin Physiological Society. This presentation that was made by Robert Koch is now regarded as one of the most influential presentations in the medical history. Paul Ehrlich, who attended the lecture, was inspired by Robert Koch's work on tubercle bacilli, and he further inspired many people to work on tubercle bacilli. After he isolated and related that the tubercle bacilli or Mycobacterium tuberculosis was responsible for tuberculosis in the humans and animals, Koch began his studies of cholera. Cholera was endemic in India and had spread to Egypt, causing fear among the Europeans of the upcoming pandemic. So the government of Germany appointed Robert Koch to lead a scientific expedition in Egypt in order to solve the problems of spread of cholera. The team consisted of 4 scientists who began their work in Alexandria. The team found that a water pump which supplied water to many parts of the particular area was responsible for the spread of the disease. So they stopped the water supply from the water pump, thus putting an end to the epidemic in Egypt. But although the epidemic ended, Robert Koch did not complete his studies to relate the causative agent of cholera and the bacterium that was present in the water supplies. So, Robert Koch traveled from Egypt to Calcutta where the disease was an endemic. So, in Calcutta, Robert Koch started examining the intestinal mucosa of the people who died because of cholera, and he observed comma-shaped structures in the intestinal mucosa which were not present in the mucosa of the normal or healthy individual. So he assumed that the comma-shaped bacillus that was present in the intestinal mucosa of infected people was the organism that was responsible for cholera. So Robert Koch used different culture media and tried to cultivate the organism on the media. After cultivating the organism on the media, Robert Koch observed that these bacteria were motile, they were aerobic and were fast growing bacteria. And because the bacteria was a fast growing bacteria, the spread of the bacteria became very easy. And cholera was restricted to the humans. Animals did not show the symptoms of cholera because all the experiments that Robert Koch performed on the isolated organisms were in animals, and inoculation of animals with the isolated bacteria failed to show the symptoms of the disease. So Robert Koch came to the conclusion that the organism that was isolated was the bacteria that was able to cause infection only in the human beings. Later, during an European outbreak of cholera, he successfully infected the guinea pigs. He applied the Koch's postulates and confirmed that the comma-shaped bacilli was the cause of cholera disease in the animals and humans. Later, Koch started studying the epidemiology of the disease. His epidemiological analysis was very precise, and he noted that there were no spontaneous epidemics outside India. The origin and recurring pandemics was in the Ganges Delta. The Ganges Brahmaputra Delta, also named Ganges Delta, the Sundarban Delta, or the Bengal Delta, is situated in Asia where Ganges and Brahmaputra rivers discharge into the Bay of Bengal. River flooding produced marshes where vegetation was abundant. Excreta from densely populated areas supplied bacteria which grew readily in the moist, fertile environment. Intestinal exposure to contaminated water caused disease in the susceptible hosts. Through excreta, the bacteria would return to the water supply. Pilgrims and travelers spread the disease throughout the country and to distant shores. So Robert Koch's discovery meant that access to clean water was necessary to prevent the spread of cholera. Thus, in Calcutta at that time, water lines were placed to circulate filtered water to the public in Calcutta, and soon it was observed that the incidence of the disease it reduced. Robert Koch's discovery was a public health victory, you can say, or triumph, which affirmed the importance of water purification and made water analysis a cornerstone of public health. He also studied malaria and established the guidelines for its prevention. He studied typhoid fever and also established the carrier state in typhoid fever. In India, he studied plague. In East Asia, he studied the sleeping sickness, and in Rhodesia, now it is South America, he studied rinderpest, malaria, sleeping sickness, horse sickness, and relapsing fever. So, a major contribution was given by Robert Koch in the field of the germ theory and his postulates, which were used for assuming that a particular bacteria is responsible for a particular disease in animals, humans, or in plants. Thank you.